My name is Reuben Shaw. I'm an assistant professor at the Salk Institute here in La Jolla. And my laboratory is interested in biochemical pathways that are deregulated in certain forms of human cancer. And we discovered a few years ago that, surprisingly, these same biochemical circuits are also deregulated in type 2 diabetes. And in particular, there's one enzyme known as AMPK that serves within the cell as a sensor of energy levels. And when energy levels are low, such as when the cell is low on food, nutrients, AMPK gets activated and serves to slow cell growth and kind of slow cellular metabolism in a way that the cell can survive and live to another day. Given these fascinating connections between metabolism and cell growth, we were very specifically interested in what some of the biochemical events that AMPK was regulating might be. Just recently, we made this exciting discovery that AMPK is directly talking to another enzyme that turns out to be kind of the watchman starting a process known as autophagy. Now, autophagy is a biological process in the cell where the cell self-cannibalizes some of its internal stores. When nutrients are low, the cell can actually break down some of its internal components to free up building blocks to be used for a number of cellular processes. So scientists had been studying autophagy and how it worked, and they knew somehow it connected to nutrient status, but no one knew how. And no one knew how to trigger it other than taking away nutrients. One of the reasons that scientists were so interested in this process of autophagy is that other than conditions when nutrients are low, it's also used all the time in cells as a kind of quality control mechanism. So the cell is constantly monitoring whether all of its internal components are in the right shape and they're in the right place and they're not aggregated. And in fact, this process whereby its internal components can aggregate and get misfolded, that goes wrong in a number of neurodegenerative diseases. It also goes wrong in a number of cancers. And finally, it goes wrong in type 2 diabetes. And this common connection between these, at face value, very different types of diseases has only been appreciated in the last 5 years. But yet, no one still knows how to artificially trigger this process to try to correct that damage. So while we were trying to investigate how AMPK might function to regulate cell growth and metabolism and who it was talking to get that done, we discovered that AMPK was talking to another enzyme called ATG1. It also has another name called ULK1. And ATG1 is the first step that basically starts that process of self-eating. So without ATG1, the cell cannot kick off this process and contribute to that quality control. So with this discovery, we now know that drugs that can activate AMPK in cells or in people can stimulate this process. This is very exciting because we already know that metformin, this widely used type 2 diabetes drug, activates AMPK. And in this study, we showed that metformin itself can trigger this switch and initiate the process of autophagy. So we're very excited about some of the ramifications of this and how we can use this connection between AMPK and ATG1 to make new compounds that might be used not only to treat diabetes, but also certain forms of cancer, as well as neurodegenerative diseases.